Good morning or good afternoon, everyone, depending on where you're joining us from, and welcome to today's webinar. My name is Eugenia, and I'll be your host. It's our pleasure to have Solvius with us today, who will be presenting this webinar titled NGS Adoption for Viral Safety from Experimental Tool to Regulatory-Endorsed Standard. Let me introduce our esteemed guest speakers for today. Donna Mahmootri, Head of Strategy, Large Molecules, and Arno Bott, a Senior Product Developer. Now I'll provide some housekeeping information before we start. First of all, I would like to welcome you to our webinar platform On24. You will notice that this webinar is browser-based, so if you disconnect for any reason, please just click on the link that you received via email to rejoin the session. During today's event, there will be plenty of time to ask questions to our speakers. You can do that via the questions widget. Just type your question in this box and click submit. We'll allocate some time at the end of the session to answer questions or any thoughts that you may have. If you require any assistance, please use the help widget, and you can move, resize, and maximize any of the windows in front of you to get a better view of the slides. But now please allow me to welcome Donna Mahmootri. Over to you.
Hi, Eugenia. Thank you very much for the kind introduction. I'm delighted to have the opportunity to talk about next generation sequencing and its application for viral safety. It's a topic that I have been passionate about since the mid-2000s, and excited to tell you a little bit about where we're going with this technology. And before we start, I just wanted to around why viral safety matters. I'm sure most of the people that join the webinar will be quite familiar with some of the challenges that have been faced in the industry, particularly throughout the early 20th century, when there were a number of incidences of infectious virus transmission from blood-based drug products and tissues, and even some vaccines. Obviously, these were widely reported, and they had significant impact to the quality of patient life. But I'm pleased to say that we have come a long way since that point in time, and we have definitely made a lot of progress in ensuring that biological drug products can be trusted. And one of the most important milestones in building that trust was a move to continuous cell culture and recombinant DNA technology for the production of biologics. And I think it really was the stringent manufacturing and quality control testing protocols that were implemented with those production methods that have effectively eliminated the transmission of active virus to patients receiving these modern biologic products. And when we talk about approaches, the robust quality by design that manufacturers have been embedding into their processes really have become that cornerstone of viral safety for decades now. And they have very much served the industry very well. The whole trilogy of prevent, detect, and remove, that cornerstone has made sure that we are preventing the introduction of viral agents into the manufacturing process through activities like screening your source materials and, where possible, eliminating the use of animal-derived products in the production process. Moving into detecting potential viral agents through the testing at various steps throughout that production process, and importantly, making sure that we are using validated and sensitive detection methods that are fit for purpose. And finally, moving towards the remove section of the cornerstone of our quality by design approach. Any potential viruses, the idea is that we are looking to remove these or inactivate them during downstream purification, with the intent that if an unfortunate adventitious agent did make its way into the process, that would be effectively removed. And collectively, these steps or this approach has successfully identified a number of contamination events during production processes, and this has happened to some of the best manufacturers out there. So while we have prevented the transmission to patients of any infectious virus agents, we do still see contamination events occur. And when they do occur, these can be quite costly, both financially and reputationally. Downtime during a manufacturing process to clean up or investigate an contamination event can be quite costly, sometimes into the millions of currency. Reputationally, of course, if there are delays to drug products reaching patients or shortages, then this can have a significant impact to both the patient and the reputation of a company. Certainly, you would expect some additional enhanced regulatory scrutiny, but also this can also impact and reduce consumer and investor confidence. So we know that the virus that they are preventing and detecting and removing viruses should they make their way into the manufacturing process. So I suppose the question for today that we want to explore is, with the recent regulatory guidance endorsing the use of next generation sequencing in viral safety testing, will we start to see this advanced solution taking a leading role and moving beyond just the adjunct or investigational tool, but really taking stage in the quality by design approach to viral safety? But we're here today because I think it will, and I certainly do believe that we are now seeing that pivotal shift towards next generation sequencing being adopted as a replacement for those traditional methods. This is intended just to take a little bit of a look back at the past 20 years in next generation sequencing. For me, I can hardly believe that it's been 20 years since this technology hit the commercial market. I was fortunate enough to be working for an organisation that was an early adopter of the methods. And back at that point in time, there certainly was a lot of interest from the biopharmaceutical industry. And many people were keen to embrace the technology as an integral part of their viral, safety characterization. And I think some would say that the biopharmaceutical industry was too slow to embrace it holistically across the viral safety testing quality by design approach. But I don't necessarily think it was slow for no reason. I would say that perhaps next generation sequencing became a victim of its own early success. Certainly early on, around the 2008 to 2010 period, there was a lot of interest and a lot of data being generated on proof of concept. Many studies were published using next generation sequencing in the biopharma industry. And I think there was a lot of excitement in the potential of that technology. And this was somewhat fueled further in 2010 when it was used to identify porcine circovirus contamination in rotavirus vaccines. I suppose the downside was that this incident really did catch manufacturers and regulators off guard. Those vaccines, they were already approved as safe, they had been widely used, and finding this adventitious viral DNA, it had gone undetected using conventional methods. So on the one hand, that single incident promoted the power of next generation sequencing, but it also highlighted the regulatory and interpretive risk of that data. Of course, after much investigation, the porcine circovirus was found not to be infectious to humans, and it did not pose any concerns or risk to health for those who had received the vaccines. But I think the regulatory and the public fallout was significant. And from my perspective, I do think that this fostered a conservatism in the adoption of NGS at that time. And so while other industries like the healthcare and the food sectors were actively adopting next generation sequencing as a standard for disease diagnostics and infectious disease surveillance, the biopharmaceutical industry became more cautious about fully embracing next generation sequencing, and I think instead focused more on establishing robust frameworks to support its application. Fast forward 15 years later to 2025, and we're now seeing regulatory authorities actively endorsing the use of next generation sequencing as part of that quality by design to viral safety. And I suppose the next session I'm going to hand over to my colleague, Arno, to talk a little bit more about those barriers to adoption and how the industry has started to overcome those challenges.
Yes, thank you, Donna, for the introduction. So as I said, I'm Arnoud, and I'm going to take you to a quick overview of what the barriers of adoption have been for NGS viral safety and what has been done in the last couple of years and what's still very much ongoing to move this forward into application. So the barriers of adoption, they can be split up into several categories. So first of all, there's technical complexity. If you think of NGS viral safety, you have to keep in mind that NGS viral safety is in essence very different from a standard NGS technology, where usually you expect to get data which aligns to the reference genome of the species you expect, and then you know your experiment worked. Of course, in this context, we're trying to prove that something is not there. And that, of course, comes with a whole set of different challenges, because you need to have certainty that if a virus would have been there, it would have been processed. So this poses all sorts of technical challenges, as in we need to define the sensitivity and, let me get my pointer, sensitivity and dynamic range. We need to monitor and understand any sample preparation biases that might exist that might cause a certain subtype of viruses to be detected less well than other viruses. Of course, as Donna mentioned, we also need to make sure that our turnarounds times are comparable to the other technologies, because producers still need to get their products to market fast enough. And then lastly, we also require a lot of expertise to perform this, because we get a lot of data, a lot of sequencing reads, and from that we need to make sense, is this a virus, or is this something just does look like a virus but is not an actual virus infection in the sample? The second category of barriers is the data analysis interpretation. So that very much coincides, of course, with expertise that we require, but the limitations of the existing bioinformatic workflows to differentiate viral signals from host or sample background sequencing reads, and the lack of consensus therein has been a major limitation into the adoption of NGS viral safety. This was also massively hampered by the lack of good, solid reference databases. And of course, what does it functionally mean if a virus nucleic acid read is detected in the sample? Does that then mean you have an active virus infection? Do you then need to do follow-up experiments? Of course, another factor that was very important in the slow uptake of NGS viral safety is the institutional inertia. There were insufficient established systems for NGS viral safety, and that did not give people confidence that this would be the right solution for their workflows. And of course, people were very scared of finding something that they didn't know was there, and that would limit their production and getting their products to market. And lastly, of course, Donna already alluded to it, the early WHO update to endorse NGS was not very clear yet in how should NGS then be applied in this workflow. And because of the lack of clarity there, as in what do we expect from these NGS workflows, where can you use them for what kind of samples, of course, made it also very difficult to establish a good, stable validation workflow and what is expected in terms of sensitivity and specificity. Okay, if we then move towards the ongoing efforts to overcome these barriers to adoption, we basically have three major initiatives that have been ongoing for the last couple of years. So we have standardization, benchmark studies, and regulatory clarity. First of all, to standardization, one major step forward is the release of the Center for Biologics Evaluation Research panel, so CBER panel, which is a panel of 5 viruses, and they have been established by a group of experts, and they contain all types of viruses that are relevant for these sorts of assays, because you can imagine there's a gigantic pile of viruses in the world. There's no way you can validate them all. So it's very good to have a standardized reference set to validate your assay on, because then if the next guy also validates their assay on this set of viruses, actually these assays can be compared. So as you can see here, this CBER panel has 5 different viruses in it. Some of these viruses are enveloped and some of these viruses are non-enveloped. That's of course very relevant for the nucleic acid isolation bit. And then if you look at the genome topology, there's some dsDNA, ssRNA, circular, larger, smaller viruses. So it's really, it's a small number of samples covering a large number of variables in terms of what can your virus look like. So this was released about 5 years ago. And then a couple of years after that, in March last year, the official WHO reference panel for adventitious virus detection was released, which is actually the same CBER panel 5 viruses, but with 2 additions added. So they added the MMV and the beta-coronavirus. So that's another ssDNA, which is always very difficult to detect just from a biochemical perspective, and another ssRNA virus, just to be a bit more solid on covering all the variables of what these viruses should look like. So this WHO panel, the 7 viruses, is now the official panel that is recommended for use during validations, and the CBER panel virus is still CBER panel is still available, and it's actually highly recommended to use that for your assay development and optimization, so that when you go into your validation with your WHO panel, you know exactly what you're gonna find. Then the next major enhancement in standardization is something I alluded to before. So the public reference genome databases, we've all used them, the GenBank, the NCBI. They're awesome databases. They have a gigantic amount of sequencing data, but the annotation is very problematic. So there's lots of viral sequences there that are not annotated as virus, and there's lots of genomic insect sequences that are annotated as virus. And of course, making sense out of all of that, if you're just analyzing next generation sequencing data, is extremely problematic. This is why it's extremely important that a couple of years ago, the RVDB, the Reference Viral Database, was released. And this is a highly curated database for viral sequences. So they have gone through all the sequences from the NCBI and GenBank, and they've sorted out, yes, this is a virus, and no, this is not a virus. And that really helps us make better sense out of NGS sequencing data that we get. Also important to note, so it's highly curated, it's not non-redundant, so you don't have all these duplicated sequences in there that cause your reads to be distributed over multiple hits. And they're still actively working on it, still being regularly updated. So making use of this RVDB database for your NGS viral safety data analysis gives you a lot more confidence that if you get a viral hit, it's actually a viral sequence and not some sort of misannotated sequence that you're looking at. Then benchmarking studies. We now have these great standards and an idea, okay, so what types of viruses to look at for a validation. With that, we've also seen more publications and method validations coming out where these viruses have been used to compare methods in different labs and just to show that the system works. This helps massively clarify what is achievable and which methods work. Especially also for the data analysis part, that's very important because there's really choices to be made in the data analysis of the NGS data that have a strong effect on what you are able to detect. Additionally, in conferences like the PDA virus meeting and conferences like this, workshops on the application of NGS and viral safety detection have been organized to get the field together to actively have this discussion about how do we apply this technology, what does it get us, and what are the limitations to what do we do with a hit. This really has helped strengthen the confidence in the technology and to get everybody's perspective and align on where this fits in the field. Similarly, there have been several position papers that, again, take a consensus of the field on how this methodology works and what the strengths and limitations are. Lastly, of course, the regulatory clarity that we have gotten in June 2024, so June last year, the ICH Q5A-R2 has come live. It was released about half a year before that, and they're pretty clear. NGS is the way forward. So there's a couple of quotes here on the slide. Just for those not familiar with ICH Q5A-R2, ICH Q5A is the guideline of viral safety evaluation on biotechnology products derived from cell lines of human or animal origin. And as you can see here in the few snippets that I took out, they clearly say that NGS encouraged as a replacement for in vivo assays. But also when we look at, for example, to PCR assays for specific viruses, they also say NGS can be used to replace the large number of PCR assays. Of course, you can imagine NGS is a major step forward over PCR because instead of just looking at a few primer binding sites having to match a virus, with NGS, you actually are able to detect the entire sequence of the virus, giving you a lot more confidence that your positive signal is actually from the virus you think it is. And one more important update in the ICH Q5A-R2 is the scope expansion to ATMPs. So it does not only apply anymore to cell-based products, but also to, for example, an AAV drug substance or a mRNA vaccine. One more thing I want to highlight here is that there's a small notation here without a head-to-head comparison. This is very important because it's very difficult to do head-to-head comparison between these different assays. They have such different characteristics, and it's very difficult to interpret this. So the regulatory agencies are quite clear that this sort of head-to-head comparison does not really add much. That does, of course, not mean that you can do whatever you want with NGS. Again, right? Use the standards that they've established and they recognize, because that also allows the regulatory agencies to compare your NGS approach with that of other labs, right? So this standardization, again, feeds in here to make a robust system. So to get back to the barriers to adoption, we started out sketching. First of all, standardization has really helped to understand sample preparation biases, the reference databases have been massively improved, actually helping us make much more sense of the NGS data we're generating, and the validation challenges that we observed previously with like, okay, so what kind of specificity do you want? What kind of sensitivity? If you use these reference standards, things can be compared to other NGS assays, and therefore it gives the regulatory bodies more confidence in interpreting your validations and results. Benchmark studies have helped a lot with understanding what's the sensitivity and dynamic range. It, of course, helps establish industry expertise. It has helped demonstrate what kind of bio-IT analysis workflows work. And just the fact that more people are doing this and not seeing a lot of positive results that make no sense, that are difficult to interpret, or that are false positives, that has also helped reduce the fear of the unknown signal that everybody is afraid of. Lastly, the regulatory clarity, of course. Right, we now have clear expectations and we have harmonized guidelines in place, and that really helps everybody in how to design their NGS assay. So as you can see, most of the barriers to adoption that we were facing a couple of years ago have been actually tackled. Now, to move a little bit more to practical applications of NGS viral safety detection. In the field, there is basically two main approaches that are being used. There's transcriptomics, where you basically just sequence all the RNA in a cell. Of course, then you're looking for viral RNA, so that's viral transcripts. And it's the best way to detect active infections. The other option is metagenomics, which is the analysis of all the nucleic acid in the cell. So that can be DNA and RNA, single strand, double strand. That, of course, then targets any viral nucleic acid. That can be a viral transcript, but can also be the viral genome of a particle not even currently inside a cell. The downside of this one is that the analysis complexity is a little bit higher because you just have a lot more nucleic acids to look at. But then the positive side of that is that it is both suitable for cell-based samples, so your show bioreactor samples, but also cell-free samples like your mRNA vaccines. Just to give a bit more context with the ability to detect all nucleic acids, at the bottom you kind of see a graphical representation of where a virus can be during an infection in a biological sample. So first of all, if we look at the top three, if a virus comes into a permissive host, it can, of course, it can infect the cells, it goes and replicates. So there's intracellular transcripts, there's intracellular viral particles, there can be particles attached to the cell surface, and of course it can secrete viral particles. But there's also situations where the virus can infect but go dormant. So think, for example, of a herpes virus. You can have proviral latency, so the virus genome can integrate in the host genome and not actively be transcribing at that time. And of course, if we go to the bottom three, we also see that there's possibility for non-permissive hosts. So say PCV1 goes into a human cell culture. In principle, that should not be infectious and replicate. So the strong side of metagenomics is that regardless of what kind of infection we're looking at, whether it's an active infection, a dormant infection, or even a non-permissive host, metagenomics casts the broadest net, and it can detect any of these viral nucleic acids present in the sample. Transcriptomics, however, it just gives you more confidence because it focuses on the transcribed RNA of the virus. It gives you confidence that the virus actually is actively infectious. but it doesn't mean it comes together with the weakness that a latent infection might be missed. Then talking about coming back to the standardization, as I mentioned before, we have the viral particle standard now, which is actually the ideal type of reference sample to use for a metagenomics approach, because they're viral particles, they're not actively infecting, so they don't have the viral transcripts in the sample. The benefits of this is that they're very easy to standardize, but of course they don't mimic an active infection in the samples. For transcriptomics approaches, infected cells are usually used for validation. So you take an infected cell line and you mix it in certain ratios with a not infected cell line, and you can determine what percentage of cells is infected. It also means that because of that difference in the way it's set up, the limits of detection are very difficult to compare. Because for transcriptomic approaches where you mix percentages infected cells with percentages uninfected cells, you can only say something about the proportion of infected cells that you're able to detect. While for metagenomics, you have a very different definition of your LOD, which is number of viral genome copies in the amount of cells that you spiked it into. So that again makes the comparison between these methods very difficult to do. When looking at how to design a NGS viral safety workflow, I'm looking specifically here at the metagenomics workflow, because that's the one where we have those standards available now. Of course, the first thing that you really have to look at in the wet lab is to make sure that all your nucleic acid is processed unbiased, right? So we have DNA, we have RNA, we have large molecules, we have small molecules. And especially at the first step, the total nucleic acid isolation or the synthesis step, those are really the steps where certain types of viruses or certain types of nucleic acid may not be processed as well as others. That, of course, leads to poor sensitivity of those viruses. So that's really something that needs to be thoroughly looked at. Then, of course, we have all sorts of types of samples and viruses that we may want to test for. So we need to make sure that the assay build we do is suitable for the type of sample that we are going to process. So if we, for example, look at a CHO bioreactor sample, it makes most sense to just take the whole bioreactor aliquot, so that's cells plus the culture media, because the viral particles might also be floating outside the cells, and process that as a whole to see what the viral load is in the overall sample. But the total nucleic acid isolation procedure you would want for that is, for example, very different for when you get an mRNA drug product. But obviously, you don't need to cell lysis there, but the viral particles still want to lyse. So it's important to also look at which protocols do you want to apply for the sample type that you're actually interested in. Second thing that's important is, like I said at the very beginning, we're trying to prove that something is not there. So we need to be very certain that we did all the steps correctly. For this, system suitability control should be added at the start. So you can monitor all the biochemical sequencing and data analysis steps to ensure that if a virus would have been there, it would have been processed, it would have been taken along through every step of your wet lab protocol and survive your data analysis. Of course, the reference panels that I just mentioned, the CBER and WHO panels, they help monitor all these steps, of course. And then for data analysis and interpretation, this is actually one of the most challenging steps. What we generally see is the increasing the stringency of the amount of viral genome that you have present actually reduces very much the amount of false positives that are reported. And automation is strongly recommended to reduce unexpected signals. For the, just to give you an example of how we build our NGS viral safety workflow, we basically incorporated most of the points above. So we have here we start on the left with the system suitability control that we spike into the sample before we start to do nucleic acid isolation. This is of course an RNA, so also during DNA synthesis needs to be processed. That shows that an RNA virus would also have progressed in the workflow. Then we go through the standard steps of NGS library preparation and sequencing, and in our data analysis, of course, we report it. And in terms of our threshold for the detection of the virus, we only report viruses as present if more than 50% of the virus genome is present. That's based on the premise that for a virus to be active and infectious, you can't only have 10% of the virus genome. You need to have the whole thing to sustain that. And as you see here, this is a aggregate of 4 independent experiments we performed on different cell lines with different operators with the CBER panel viruses. And you see here that at our limit of detection of 1,000 viral genome copies in 500,000 cells, we very neatly get about 60% of the virus genome covered. So we have a very stable and robust detection of all the viruses of this model set. Of course, that's all great in a lab flow, but everybody only cares about what does it actually do on our customer samples. So luckily, we were able to do a beta project with Beyond This, a antibody-drug conjugate company here in the Netherlands. And they were willing to share a CHO bioreactor sample with us, so one of their antibody-producing lines, a 12-day bioreactor bulk harvest sample. And yeah, it's a little bit uninteresting to show that we didn't detect any virus. But of course, we do have a lot of good results here because everything was exactly the way we intended it to be. We detected the system suitability control, you can see here in the table, at 99% and 97% in all the test sample replicates and the negative control. So the workflow worked brilliantly. And of course, most importantly, we didn't see any unexpected virus, neither in the customer sample, but also not in negative control. So this is of course perfect. However, we were not completely satisfied yet because we wanted to make sure, of course, that if a virus would have been there, it would have been detected. So for that, we needed a confirmation test. Where basically we took the same sample, we processed it, of course, in exactly the same way, but before we started it, we added some of the CBER reference virus panel at the LOD I mentioned before of 1,000 genome copies per 500,000 cells. And then we saw all the viruses very nicely showing up. You see here in the bar plot that all the viruses were again at 60% or higher. So very clearly detected. You see here for the feline leukemia virus and the respiratory syncytial virus that we have a very, we don't have one specific spike of virus. We detected the whole thing is there. That clearly shows that this is that gives us confidence that it's a true positive hit. That being said, I would like to hand over back to Donna to talk about NGS adoption.
Well, thank you, Arno, for taking us through the barriers to adoption and the approaches to developing these types of next generation sequencing methods. And a big thank you to Beyondis for supporting our validation by sharing some real world samples. It certainly makes all the difference to have that collaboration with our customers. So a big thank you to them. Okay, so where does that leave us today? Well, certainly we are, in my opinion, seeing that pivotal shift in NGS adoption for viral safety. Certainly, we've seen an increasing demand for this tool as part of viral safety testing strategies. Now, we have just heard from Arno on the drivers and the catalysts for acceptance. Obviously, the regulatory authorities endorsing this method has been a great help. The published validation pathways that we've seen from industry peers and the rising, or should I say, costs of next generation sequencing have certainly helped. And of course, the improved bioinformatics capabilities. But I think the right tools and the right framework for applying those tools is great. But one of the other drivers that we see is the adoption, or the modality-driven adoption of this tool. Certainly, from our internal perspective, informed from voice of the customer and customer feedback, this indicates that the demand is proportionally higher in newer modalities like your cell and gene therapy vectors. Certainly where product complexity is high and the downstream processing to remove or inactivate infectious viral agents is low, we're seeing next generation sequencing become an expected standard to assured integrity and safety. And so the table here really is some internal trends from our perspective on the rate of adoption and some of the key drivers or motivators behind why these particular modality producers are approaching us next generation sequencing services. Clearly with the cell and gene therapy market, many of these products have viral vector backbones. That makes the use of a broad, unbiased detection method essential. But we are seeing interest in looking for replication competent viruses within the vectors themselves, as well as this unbiased detection for contaminating viruses. When it comes to the cell therapies, clearly some of the interest points here are really around the likelihood of latent or integrated viral sequences that could cause reactivation later in use. And when we start to look at the more traditional or classical biologics, your recombinant protein products and your monoclonal antibodies, certainly we're seeing a slower uptake or a slower demand for these services. Historically, these are well-characterized cell lines that are used to produce these products. And so there is a kind of known quantity there. The viral risks are typically expected to be low. So where we do see these methods being adopted, it really is around replacement of in vitro methods and moving into the 3 Rs and sustainability. And vaccine producers, for many years have been performing next generation sequencing in-house, and we continue to see a trend and an endorsement of using next generation sequencing as a quality control tool, really for root cause readiness. But the emerging field, I think, where we are seeing a lot of interest is in these novel modalities like oncolytic viruses and exosomes. And I think the primary driver there is really the lack of, identified risks with those types of modalities. And so comprehensive characterization and inquiries about how we can help those manufacturers characterize from preclinical to throughout their production processes is common. So in terms of adoption, yes, I think we are seeing that pivotal shift from investigational tool to a staple in that viral safety approach. So, like all fields, it continues to progress. There's a lot of work actively going on in the field to continue to advance and improve the use of next generation sequencing technologies. Certainly an area that we're keen to embrace is long read sequencing and some of the complementary benefits that type of analysis has in the viral safety landscape. One of the things that I'm keen to see, and I know there's a lot of work going on there, is the development of standards for the transcriptomics approach to next generation sequencing. So a lot of great work happening in the field there, and I'm keen to see what comes next for that. looking more broadly, I do believe that we will start to see next generation sequencing as the favoured approach, replacing those in vitros ultimately as part of the QC workflow for advanced therapies. And we're already hearing a lot of noise in the industry from early adopters using AI-based tools to help with the analytics and hopefully reduce the likelihood of those false positives. So that's a very exciting field. There's a lot of buzz in the industry there. So I look forward to hearing a lot more about that. I think that's the next frontier of how we get regulatory support and adoption for the application of these tools in analytics. And of course, integrating the use of next generation sequencing with proteomics and bringing those two worlds of genomics and proteomics together, certainly advancing well. And I look forward to seeing that integrated into quality control processes more in the future. So the take-home message from us, the momentum is here, the shift has started, and we're very excited to be part of that journey. For next generation sequencing and viral safety and the new modalities coming on, I think the use of next generation sequencing will certainly help ensure that those next level of advanced biological therapies are as trusted as the traditional molecules. I think that's all from us. So thank you very much for attending.
And thank you very much for your presentation. Just a reminder to the audience to ask questions. You can send them via the questions chat. Just type them in this box and click submit. Please note that you can still send your questions after the webinar and they will be answered at a later date. And it looks like we've got several questions already. And here's the first one. How to deal with host construct-derived sequences?
Arno, would you like to take that one?
Yeah, absolutely. That's a very interesting question. Yeah, so of course, if we, for example, look at the CHO bioreactor sample expressing antibodies, we all know they use the construct in there with a CMV promoter and a poly-A tail from SC40, right? So there's always viral elements in there. An approach that we specifically took, we already know they won't pop up because the SV40 promoter is not 50% of the CMV promoter is not 50% of the CMV genome, right? So this sort of stringency already helps you filter it out. Of course, you can also do an a priori filter on bioinformatically removing these constructs. So both approaches are perfectly fine. I think I would also like to add here that during, of course, your assay development, you can also already test all your model organisms that you're going to receive to ensure you can already remove any signals that are always in a CHO sample, for example. So you can, during your assay development and characterization for each genome that you expect to test, make sure that you already remove these signals to avoid complications later on.
Yeah, thank you very much for that response. And we have another question from the audience. Is there a difference in sensitivity between metagenomics and transcriptomics?
I think I already alluded to that a little bit earlier during my presentation. So because of the differences of the approaches, for metagenomics, we calibrate or validate our method and our limit of detection based on viral genome copies. And for transcriptomics, you usually use a mixture of infected versus non-infected cells. So the absolute numbers there are very difficult to compare. What I can say is that, of course, an active infection where viral transcripts are present that's a lot of nucleic acid that's detectable, right? So that's, of course, what transcriptomics is built on. At the same time, for the metagenomics approach, these RNAs are also processed, so we also detect those. So in terms of nucleic acid that's being yeah, that's detectable. That's probably comparable. It's just that the numbers are difficult to compare.
Yeah, thank you very much. And we have another question. Is metagenomics always the preferred choice, or are there situations where transcriptomics is clearly better?
I mean, I can chip in. Arno, if you want to add to it, please do. I think both methods have their benefits and limitations. Certainly, as we mentioned earlier, metagenomics definitely is a broad spectrum catch-all net, very appropriate for looking for unknowns. Certainly, I think that's its preferred application might be in a broad screening test. Transcriptomics, obviously, looking for active or indications of active infection is definitely a strength for that technology. I personally wouldn't say that we have favour one over the other. I think quality by design approach to viral safety. My personal opinion is that I would like to see us use metagenomics as a broad spectrum first, test that we use to look for any potential unknowns or known viruses, and then use the transcriptomics as a follow-up test should we actually find any signals to further interrogate that data. Arno, anything else to add?
I think that sums it up very nicely. Maybe the only thing to just add to that is there are, especially in the last year, there were some concern about the dormant infections. Of course, that's where metagenomics really, but metagenomics can detect those. Transcriptomics only, well, of course, if the dormant infection becomes active again, transcriptomics also detects it, but there is the way out for the virus to hide in the host genome to evade detection. So that's where metagenomics, cannot be fooled, if you can put it like that.
Yeah, thank you very much for that response. And we have another question from the audience. What is the typical turnaround time for an NGS viral safety study from sample receipt to final report?
Well, I think different providers of that service probably have different timelines. But for me, if we talk about our own service and our aspirations for turnaround time of that service, then certainly 14 days is a good aspirational timeline for us to target. We would like our service to be competitive with some of the other alternative technologies on the market. So typically from receiving a sample and initiating testing to completing the analysis and having a certificate of analysis, just a pass-fail report, then 14 days is certainly a good target timeline for that type of service.
Thank you very much for that response. And here's our next question. If we want to transition from a traditional panel of PCR assays to an NGS-based approach, what would a realistic migration plan look like?
I can talk a little bit to that. I think it really depends on how those tests are being used in the customer's quality by design approach. Certainly the regulations indicate that we do not need to do head-to-head studies, and that's the official party line, and that's perfectly acceptable. And as long as the manufacturer is aligned with the regulators on changing from one technology to another, then there should be no problem. Typically what we find is that our customers like to do some suitability testing up front just to build confidence internally in the application. Not a head-to-head study, but certainly a system suitability. They like to understand the validation behind our services, and we like to support them with data around our validation so that it makes their change of methods quick and easy. But certainly the trends that we're seeing are that customers prefer to do an upfront suitability test using our methodology before making those decisions to replace or add the next generation sequencing methods to their portfolio of tests.
Yeah, thank you very much. And just a reminder that you can still send any questions while watching on demand. All questions will be answered at a later date. I would like to thank the speakers for what was a great presentation and Solvias for sponsoring this session. To that in this, you will receive an email telling you how you can access the on-demand version of this webinar. Alternatively, you can access it through our website, which is pmi-live.com. We look forward to sharing further webinars with you, so please do keep an eye on the website just mentioned. Thank you once again, and I hope you all have a nice day.